Okay, we should be good now.
All right, welcome everybody. It's a pleasure for me to introduce Luke Fenlon for his thesis defense. Luke's an MD PhD student, joined the microbiology department in 2010. He graduated from University of Wisconsin, where he worked for Diana Downs as an undergraduate. He's an author on 2 papers resulting from his work there. He also got several research awards, including a summer fellowship from the American Society for Microbiology. Luke had a challenging project aimed at discovering the targets of phagocytic superoxide. So it's been known for over 100 years that phagocytes engulf bacteria and kill them, but the mechanism by which they actually cause the death of the organism is really still unknown. And over the last number of years, we've shown that the dogma of killing by, for example, causing oxidative DNA damage does not seem to be correct. And rather, our data suggests that the targets of phagocytic superoxide in bacteria are outside of the bacterial cytoplasm. And the problem is we don't have no clue what those targets are. So we've been thinking a long time about how to address that problem, but we needed 2 different things to happen. First, we needed the technology to catch up to make the experiment doable. And secondly, and more importantly, I needed a student with the brains, fortitude, and work ethic to actually pull off the experiment. And Luke was that student. So he'll describe his what we call differential TN-seq experiment to identify mutants of Salmonella that affect virulence in our mouse model and genetically interact with SOD-C, which encodes the periplasmic superoxide dismutase, which we know is important for combating phagocytic superoxide. I just want to emphasize that this was a huge experiment, and there wasn't a single step in this process that was trivial. But Luke diligently and meticulously worked through every parameter. This includes numerous control experiments to ensure that we understood where the bottlenecks were in this system. In other words, where mutant Salmonella would drop out by chance instead of for biological reasons. He isolated 90,000 transposon insertion mutations, picking each one individually into microtiter wells, although he did use a robot for that. But he then did infections in well over 300 mice. And it took really 3 years of careful planning and control experiments to really do this correctly. It was just phenomenal. The last step in this process was the DNA sequencing. And it turns out that there was some artifact in this process that we really don't understand. But more recently, Yumi Iwadare in the lab, with the help of Paul Jensen, has developed new methods and resequenced Luke's experiment. The reason I'm saying that is that although Luke's been out of the lab for a couple years in medical school, we're just now getting to the point where we can really analyze the results of his experiment. And I expect the follow-up experiments to take a number of years. Luke also worked on other projects, and he'll talk about that too. So Luke has all the traits we look for in a great graduate student and scientist. He works very hard. He was always at his desk when I arrived in the morning, even if that was at 7:30 in the morning. He was there when I left in the evening. He was either, working at the bench or in front of his computer. He's a very careful experimentalist. We thought hard about all the results. He's also a great citizen. He actively participated in the graduate program, the infection biology training grant program, the medical scholars program, and the medical school activities at large. As an MSP student, he's also had to balance graduate school and medical school. He performed excellently in both arenas. He just graduated with his MD, and he will begin his residency in internal medicine at the University of Utah in a month or so. So I look forward to his future successes. With that, I'll turn it over to Luke and let him present his research.
All right, thank you, Jim, for that very kind introduction. Let me get my slides pulled up here. All right, hopefully you can all see that now. And so just, um, I'm excited to be able to share some of my work with you today. Thank you all for joining me virtually. Today I'm going to talk about some of the work towards my dissertation entitled Survival of Salmonella in the Macrophage Phagosome. And so to begin this talk, I'm first going to give an introduction into the favorite pathogen of the Slough lab, which is Salmonella, and then talk about a story that I worked on looking at how SOD1 acquires copper in the periplasm, then talk about the project that Jim just explained, trying to identify the target of phagocytic superoxide, and then finally wrap up with some conclusions and future directions. So in the Slough lab, we study Salmonella pathogenesis, and many of you already know that Salmonella is a major foodborne pathogen. Salmonella disease can be broadly thought of as typhoidal versus non-typhoidal disease. In our lab, we study a non-typhoidal Salmonella species. Non-typhoidal Salmonella typically causes a self-limiting gastroenteritis and has been estimated to cause 93 million cases and 150,000 plus deaths worldwide annually. And there's a large disease burden even within the United States. So non-typhoidal salmonella is the leading cause of bacterial foodborne disease hospitalizations and deaths within the US. Now, in certain cases, in people that have, for some reason, a compromised immune system, non-typhoidal salmonella can actually cause invasive systemic disease. And it's estimated that there's 3.4 million cases of invasive disease annually. And the case fatality rate for this can be as high as 20%. So it can be very serious for those that get invasive disease. And we know from a lot of previous work in Salmonella that specialized virulence factors are required to cause systemic disease. And one of those, the key aspects is the ability of Salmonella to survive within phagocytes, a specific type of immune cell. Here on the right, I'm showing a scanning EM of Salmonella infecting some human cells in tissue culture. So to walk through the course of infection, Salmonella is typically acquired via consumption of contaminated food or water. It's able to survive until it reaches the distal portion of the small intestine, the ileum. At this point, Salmonella preferentially targets M cells that are found in Peyer's patches in this part of the intestine, and where environmental signals induce expression of a type 3 secretion system in Salmonella that results in injection of effector proteins into these host cells. This results in membrane ruffling and uptake of the bacteria, and the immune response to this infection causes the typical inflammatory diarrhea symptoms we associate with Salmonella infection. Now, after breaching this gut epithelial barrier, Salmonella encounters the underlying immune cells. These include macrophages and neutrophils, which can take up Salmonella via phagocytosis. At that point, Salmonella resides inside the macrophage where it's actually able to survive. I'll talk more about this interaction on the next few slides, but to finish the course of infection, Salmonella then uses macrophages as a vehicle to be delivered to other organs in the body, including the spleen and liver, and in severe cases will result in septic shock for those hosts. And so for my research, I'm interested in how Salmonella is able to survive inside macrophages, which is typically a hostile environment to bacteria. So to explain that interaction in more detail, when a macrophage encounters a bacteria, it's taken up via phagocytosis. A variety of antimicrobial products are then delivered to that vacuole containing the bacterium. I'll talk more about those specifically on the next slide. Those antimicrobial factors work together to kill the invading bacterium and can go on, the immune cell can go on to generate an immune response. When Salmonella finds itself in this initial phagocytic environment, there's a two-component system, PO-PQ, that senses that environment and induces expression of an additional type 3 secretion system. This one encoded on Salmonella pathogenicity island 2. This results in injection of effector proteins into the macrophage, and that alters this normal trafficking pathway and creates what's known as the unique Salmonella-containing vacuole. And we know from previous research that Salmonella is actually able to survive and replicate in this vacuole. Nonetheless, Salmonella requires specific defense mechanisms against the host antimicrobial factors. And some of those antimicrobial factors include reactive oxygen species, reactive nitrogen species, antimicrobial peptides, proteases, and acidification of this vacuole. For my talk, I'd like to focus a little bit more on two of those, reactive oxygen and reactive nitrogen species. Reactive nitrogen species are generated by the inducible nitric oxide synthase. This is an enzyme complex that assembles on this vacuole membrane and generates high levels of nitric oxide within this vacuole. Nitric oxide itself is a potent inhibitor of respiration and can go on to form other downstream reactive nitrogen intermediates. Reactive oxygen species are generated by the NADPH-dependent phagocytic oxidase, which again is an enzyme complex that will assemble on the membrane and generate high levels of superoxide. Superoxide itself can damage specific targets and can be converted into other downstream reactive oxygen species, including hydrogen peroxide and hydroxyl radical. And in some cases, superoxide and nitric oxide can combine to form the highly toxic peroxynitrate. For my research, I'm interested in understanding more about how Salmonella is specifically able to defend against superoxide. The strain of Salmonella we're working with is Salmonella typhimurium, and the enzyme that Salmonella uses to defend against superoxide are superoxide dismutases, which I'll refer to as SOD for short, and they catalyze the reactions shown here, taking 2 molecules of superoxide and converting that into hydrogen peroxide and oxygen. So our strain of Salmonella actually has 4 different SOD enzymes, and there's different families of SOD enzymes, and each one uses a different metal cofactor to catalyze this reaction. So SOD A and SOD B are both found in the cytoplasm of Salmonella, and they've been shown to be important for defending against endogenously generated superoxide. This can happen just adventitiously during aerobic metabolism. SOD-C1 and SOD-C2 are both copper-zinc cofactor superoxide dismutases and are found within the periplasm. And from a lot of previous research in our lab and others, we know that SOD-C1 is specifically important for protecting Salmonella from phagocytic superoxide. If we knock out SOD-C1 in Salmonella, it's 7 to 10-fold attenuated relative to the wild type strain. So first, I'm going to tell you a story about some work that I did to try to understand how Salmonella acquires copper as a cofactor, or how SOD-C1 acquires copper as a cofactor in the periplasm of Salmonella. So to start out this story, I first need to introduce to you copper trafficking in our strain of Salmonella. So copper is a really interesting metal. It's an essential cofactor for certain enzymes, including SOD-C1 and SOD-C2. However, in Salmonella and other bacteria, it's known that if copper accumulates in the cytoplasm, it can actually be toxic and inhibit growth. And so bacteria have this delicate balance where they need to acquire sufficient copper to activate their enzymes, but also have systems in place to detoxify copper from the cytoplasm to prevent stress. And so here I'm showing copper 1 and copper 2. So copper can gain access to the cytoplasm of Salmonella through non-specific mechanisms. At that point, it's recognized by 2 different transcriptional regulators, QR or GOL-S. When bound by copper, those regulators activate transcription of their operons, including COP-A, QO, QP, and GOL-T and GOL-B. So GOL-T and COP-A are both P1B type ATPases that pump copper from the cytoplasm into the periplasm. Previous research studying these transporters has shown that they're functionally redundant in the purpose of cytoplasmic copper export. So insertions, if we knock out one of the 2 transporters, Salmonella still grows fine, but if we knock out both transporters, those strains of Salmonella have increased sensitivity to growth in the presence of copper. QP is a periplasmic copper binding protein, and QO is a multi-copper oxidase that can convert copper 1 to copper 2. A publication came out several years ago that caught my attention, and in this publication, what they showed was that they were looking specifically at SOD-C2 enzymatic activity and how SOD-C2 obtains copper, and their data suggests that copper is delivered to SOD-C2 from QP after it's exported from the cytoplasm by either COP-A or GOL-T. So initially, this seemed a little bit circuitous, that copper gets and crosses the periplasm, enters the cytoplasm, and then gets trafficked out one of these 2 transporters to QP and then delivered to SOD-C2 for enzymatic activity, as opposed to some alternative mechanism where SOD-C1 and SOD-C2 might acquire copper as it enters the periplasm. So we wanted to investigate this a little bit further. Since they didn't look at SOD-C1, we wanted to ask if this copper detoxification and trafficking pathway could also contribute to activation of SOD-C1. And then we wanted to take this one step further. In our lab, we really like to do virulence assays in our mouse infection model, and we wanted to ask if these mutants had any specific effect on virulence in our strain of Salmonella. So the first thing that I wanted to do was to make mutations in COP-A, GOL-T, and QP, and just confirm the previous phenotype that the COP-A, GOL-T double mutant has increased sensitivity to growth in the presence of copper. So what I'm showing you here are growth curves, just monitoring the OD450 over time. And in this one, we're looking at growth in M9 minimal media with glucose and no copper added. You can see wild type, a COP-A, QT, GOL-P single mutants, the double mutant and the triple mutant all grow similar to wild type. If we add as little as 0.5 micromolar copper sulfate to that medium, you now see that the COP-A, GOL-T double mutant and the triple mutant don't grow, whereas the single mutants grow similar to wild type again. So this confirms the previously published reports that COP-A and GOL-T are functionally redundant in protecting the cytoplasm from copper stress, and also confirms that the strains and the mutations that we made are sensitive if we knock out both of these transporters. The next step in these experiments was to actually assay SOD activity. So for these experiments, we used the xanthine oxidase cytochrome C method for assaying superoxide dismutase. And the way I'm going to present the data to you for these experiments is the ratio of the fraction of initial active enzyme to the total amount of enzyme present. And the way that I calculated that was I grew up the strains, usually either wild type or our triple mutant strains. I then isolated extracts containing our superoxide dismutase of interest. I then did initial SOD assays to calculate the amount of activity in that extract. And we know from previous research that we can add copper to those extracts, and any enzyme present that doesn't have copper in the active site can be remetylated and activated. We can then re-assay this extract to find out how much total SOD was present in that extract. And so if we look at those ratios and we compare wild type to a triple mutant, looking at SOD-C2 activity, we see indeed, similar to the previous reports, in the absence of COP-A, GOL-T, and QP, there's a reduced initial fraction of activity in SOD-C2 in that background relative to wild type. What's interesting about this data is that even in the absence of copper detoxification in QP, we still see a little over 50% of the enzyme is active. And so we wanted to see if we could exacerbate this phenotype by growing these strains in the presence of a copper chelator. And so what I'm showing here is increasing concentrations, 10, 20, and 30 micromolar of a copper chelator. And then again, looking at SOD-C2 activity of wild type compared to the triple mutant. And you can see that as we increase concentration of chelator in the growth medium, we get a decreasing amount of initial SOD activity, but it's similar in both the wild type and the triple mutant background. And if you look and compare individually each of these, we see there's about 50% less activity of SOD-C2 initially in the absence of COP-A, GOL-T, and QP. Next, we wanted to look at if SOD-C1 could also potentially be activated by this trafficking pathway. And so in panel C here, I'm showing you again SOD-C2 similar to before. And here is the data for SOD-C1. So comparing wild type to our triple mutant background, we see that indeed for SOD-C1 as well, it appears cytoplasmic copper detoxification and QP can contribute to activating SOD-C1. What's notable about this data is comparing it to SOD-C1, we see a higher initial fraction of active enzyme in SOD-C1 compared to SOD-C2. And this actually fits with previously published data based on other research. We know that SOD-C1 actually has a slightly higher affinity for copper than SOD-C2. So it's not necessarily surprising that we would see higher initial activity for SOD-C1 compared to SOD-C2. Now that we had looked at these in vitro activation assays, we wanted to take the next step and actually look at the systemic infection model. And so the way that we do that is using my favorite experiment in the lab, which is the competition assay. And the way this assay works is we take wild type and a mutant strain, we mix them in a 50-50 ratio. We then infect that population into our animal model. There's two ways we can do that. We can do oral inoculation, or we can inject bacteria directly into the intraperitoneal cavity. For all of the competition assays I'm going to talk about today, I'm doing the intraperitoneal inoculations. And the reason we do this is because this way we can ask directly what's the effect of these mutations on the systemic phase of infection. If we started with oral infection, we would encounter bottlenecks during invasion of the gut epithelium. So this is a more direct measure of systemic infection. After several days, I removed the spleen from these animals, and then one of our strains will contain an antibiotic resistance marker to allow us to calculate the ratio of the mutants in the output population. We can then compare the percent mutant output to the percent mutant input, and that gives us our competitive index. So if that value is close to 1, that means our mutant was equally virulent to wild type. If that value is significantly less than 1, that means our mutant had some sort of virulence defect. And if that value is significantly greater than 1, that mutant was more virulent than the wild type strain. So here's the results for some of our experiments for these specific mutants. And so the first one I'm showing you is just our control experiment. So if we knock out SOD1 and do a competition against wild type, we see the median competitive index is about 0.15. This corresponds to a fold attenuation of close to 7. And this experiment was done in 4 mice and was statistically significant. So this is the phenotype we typically see for a SOD1 mutant, around 6 to 10 fold attenuated relative to wild type. Now, what we would expect is if we knock out QP, COP-A, GOL-T, and any combination of these, if that trafficking pathway is important for activating SOD1 for infection, we would expect to see a virulence defect in those mutants compared to wild type. And when we look at the data for that, we see no significant phenotype. So our conclusion we draw from this is that SOD1 is sufficiently active in the absence of additional copper trafficking activation to contribute to full virulence in our animal model. Now, another way we can ask this question to further support our interpretation of the previous set of experiments is to look at the phenotype of a SOD1 mutant in competition against these mutant backgrounds. The idea behind this set of experiments is that if QP and the transporters are important for activating SOD1, we'd expect that in the absence of that transport system, that the SOD1 phenotype would be decreased because it had decreased activity, and so the virulence defect would essentially go down. And if we knock out SOD1 in those mutant backgrounds, we still see that a SOD1 mutant is statistically attenuated relative to the parent strain. So again, this suggests that SOD1 is sufficiently active in that background to be fully virulent. Now, I want to go back and talk about one other hypothesis we were able to test with this data that I've presented to you thus far. And so I need to explain one more thing that I didn't mention initially. So this part of the model is the same that I presented to you before, but now I've added the Salmonella containing vacuole membrane. And what's interesting is that it's been shown that macrophages in response to bacterial infection will induce expression of a transporter, ATP7A. This transporter inserts in the Phagosome membrane or the Salmonella containing vacuole membrane and actually pumps copper into that vacuole. And based on what I mentioned about copper potentially being growth inhibitory to bacteria, the idea is that by increasing copper in this vacuole, it can be another antimicrobial defense for host immune cells. And it's been shown in some pathogens that in certain pathogens, if you knock out whatever the pathogen's cytoplasmic copper detoxification system is, those pathogens are less virulent. The data for Salmonella has been a little bit mixed. So we wanted to ask with our strains and our mutants, which I've shown you previously in minimal media with as little as 0.5 micromolar copper sulfate, our double transporter mutant does not grow compared to wild type. So we'd expect that we could look at the competition data from these mutants compared to wild type and ask if Salmonella is experiencing significant cytoplasmic copper stress. So this is the same competition data I've shown you previously, but now thinking about this in light of this new hypothesis I just mentioned, again, we see that even in the triple mutant scenario where we expect the most severe phenotype, we don't see any significant attenuation for that mutant. So we can conclude that under our infection model with the strain Salmonella, we don't see any significant virulence defect, suggesting there's cytoplasmic copper toxicity in this infection system. And so just to summarize what I've talked about in this part of my talk, I've shown that both SOD2 and SOD1 can be activated by copper that's transported via COP-A or GOL-T to QP, and that can increase enzyme activity. However, a significant portion of the enzyme is active independent of that pathway through some other non-specified method. And we know that sufficient SOD1 is active in that absence of this transport system to be fully virulent in an infection model. And Salmonella does not seem to be experiencing significant cytoplasmic copper stress during infection. Next, I'd like to transition into talking about the project that Jim introduced in his introduction, where I did some work trying to identify the target of phagocytic superoxide in Salmonella. So again, just to set the stage for this portion of my talk, based on a lot of previous research in our labs and others, we know that SOD1 specifically defends against superoxide generated in the phagosome. And the question has been, what is the target of phagocytic superoxide, or what does it damage in the absence of SOD1? And so Maureen, a previous graduate student in our lab, published a paper just prior to when I joined the lab, where she did this really elegant genetic interaction experiments. And she concluded that SOD1 functions genetically independent of SOD-A and SOD-B. And what we conclude from that is the target of phagocytic superoxide is extracytoplasmic. And so that leaves potentially something in the periplasm or the membrane. And since what's known about reactive oxygen species biology, there aren't any obvious targets in the periplasm, we decided to take a global approach to try to identify novel genetic interactions with SOD1 using differential T-and-seq, and complemented that with an RNA-seq experiment. So to explain the differential T-and-seq experiment, first I'll walk through the basic idea of T-and-seq. I know many of you are already familiar with this. This has become a much more common experiment in bacteria. It's important to understand to set the stage for the differential T-and-seq. And so the way this experiment works is a pool of random transposon mutants are generated in a strain of interest. That pool of mutants are then passaged through some test condition. In our case, this is our mouse infection model. The most robust phenotype we have for a SOD1 mutant is our in vivo infection. Unfortunately, there's no in vitro conditions that can replicate that. That might be more high throughput. So I'll talk about some of the limitations and the challenges we face using that as our selection system. And then after that, you can collect the output mutants that survive your test condition. And then using sequencing, you can compare the input mutants to the output mutants. And so in this example, we see that insertions in gene B are identified in the input, but not the output. So that tells us that the function of gene B is important under our test condition, in this case, the mouse infection. So again, this is not necessarily a unique experiment. It's been done in Salmonella and many other bacteria at this point. What was unique about our experiment is that we did this in both a wild type and a SOD1 mutant background, with the idea that this could be an unbiased genome-wide approach to identification of genetic interactions with SOD1. I'd just like to explain in a little bit more detail about how genetic interactions can give us insight to the target of phagocytic superoxide. So on the left here, I'm showing a synthetic interaction. In this hypothetical example, if protein D and SOD1 both contribute to defense against phagocytic superoxide, you can imagine in a wild type background, if you have insertions in protein D, SOD1 is still present and functioning in this defense mechanism. So insertions in protein D are going to be represented in the output because we still have defense against superoxide. Now, if we think about the scenario of a double mutant background, we no longer have this function of defense against superoxide in the absence of SOD and protein D. So this would be represented in the data by decreased abundance or absence of insertion D in our output population in the SOD mutant background. So that's an example of an interpretation of the synthetic interaction. Another common interaction we might think about are epistatic interactions. So in this example, I'm showing protein C, which under a wild type scenario, SOD1 protects the function of protein C from superoxide, which could be either normal physiology or involved in virulence. So in the wild type scenario, if we have insertions in protein C, we're losing this normal function, and those mutants are going to be attenuated relative to the rest of the population. So that'll present as decreased abundance or absence of insertions in gene C in our output data. And if we consider the hypothetical SOD scenario, in the absence of SOD, superoxide is going to inhibit protein C. So if we add additional insertions into that gene, it won't affect the fitness of that strain relative to the rest of the population. So that's represented as insertions in the output that are relatively equally abundant to the input. Those are two examples that we might see in the data, and I'll come back to that a little bit more later. So as Jim mentioned, this turned out to be a very large experiment that took many, many months to years to initiate, and we put a lot of thought into how we managed all the controls and all the experimental conditions that we did to try to make sure we did each step as thoroughly and thoughtfully as we could. I'd just like to summarize a lot of that work with these few bullet points that I have here. So first was, how do we generate these input pools of mutants? And we determined that if we looked at about 40,000 total mutants in each background, that would give us a high level of confidence that we would have anywhere between 2 and 10 insertions in all non-essential genes throughout the genome. And we generated these mutants using a mating protocol that was developed in the Metcalf lab, in which a transposon is inserted randomly throughout the genome. And then the next big thing we needed to figure out before we could conduct this experiment was, what are our infection conditions going to be for our selection? So you can imagine that if you inject too many Salmonella into a mouse model, you're going to completely overwhelm the immune system and you're not going to get biologically relevant results. And so we wanted to figure out what size of inoculum we could use in these experiments. And we also were concerned about the population complexity. Again, as Jim mentioned, we were worried that we didn't want these mutants to run into any significant bottlenecks throughout the course of infection. And so we did some preliminary experiments and we created this unique 4-way competition assay in which we took a wild type strain and 3 mutants with known phenotypes. We mixed those together, used different inoculum sizes, different combinations of total copy number of each of the mutants, and then asked under those infection conditions, do we get output results that match what we would expect for those known phenotypes? And the summary from that data was that we used an inoculum of around 50,000 total bacteria with a population complexity of 1 to 1,000, which means each mutant present in that inoculum had about 50 copies. And we also tested a couple of different mouse genotypes to see if that significantly affected either of these parameters. And the take home was that we did this experiment in valid C mice. So based on this limitation of a population complexity of about 1,000 mutants, we took that total number of 40,000 and decided to make 40 independent pools in each background. And then as Jim mentioned, we took advantage of a colony picking robot, thanks to the MMG team over at the IGB, in which the robot can pick individual colonies off the mating selection plate and inoculate that into 384 well plates. So we decided to use 384 well plates to make an individual pool, which put us in the neighborhood of around 45,000 mutants in a wild type and a SOD1 mutant background. So once I generated all of those mutant pools, the next step was actually to carry out our mouse infection. So here, again, I'm showing just the strategy for that. We put a lot of thought into how we did each one of these individual steps to ensure we didn't introduce any bias along the way. So each pool was grown up overnight in the lab. I then infected that pool into 4 different mice. In the end, I'll show on the next slide, we end up only using 3 out of the 4 populations isolated from these mice. And the reason we started with 4 is because we do see some variability with an animal model. And so that was just another level of control where we could try to monitor the progress of this experiment. So after 72 hours, I would remove the spleen from these animals, isolate the bacterial cells from that mixed tissue. And then for each individual population from an individual mouse, I would determine the CFU per mil and then store that in the freezer. And then right before I initiated these infections, I would spike in a SOD1 mutant as a control. And the reason I did this is because when we isolated the bacterial cells, I could calculate the competitive index for SOD1 and see if it fit within the realm of what we would expect for a known SOD1 mutant. And if it did, that suggested to us that population didn't encounter any significant bottlenecks, but occasionally we would see significant attenuation, suggesting significant bottlenecks, or in some cases we wouldn't see attenuation for the SOD1 control, suggesting there was not sufficient selection for that population in vivo. So that was another one of the controls that we used. So after I finished infections for all 40 pools in the wild type and SOD1 background, which was over 320 mice later, we were now ready to combine these groups together and actually do our sequencing. And so, as I mentioned, we used 3 populations out of the 4 mice to use in our final output groups. And we carefully thought about how we assigned each population to each group to control for the total number of bacteria in our final output groups. So for each background, we had 3 final output groups, each containing representative bacteria from pools 1 through 40. And we basically could consider these our biological replicates. Unfortunately, there was not enough cells to isolate sufficient DNA to make a quality sequencing library. So we actually took these groups and grew them in a lab for several generations to reach an OD of 1, and at that point isolated DNA. I did the same thing for the input pools, again, equally combining all the pools together. And for consistency, we diluted this population, drew it under the same conditions as the output, and then isolated DNA. So in total, we submitted 8 groups for sequencing, both the individual input groups and then 3 output groups in each background. And the way that the sequencing process works is the library preparation selects for fragments of genomic DNA that contain the transposon insertion and the adjacent DNA, and the actual sequencing primer itself binds 10 nucleotides upstream of the end of the transposon, and then reads out into the genomic DNA up to 100 nucleotides. So this gives us the exact insertion location of the transposon within the genome. One way we quickly tested the results when we got all this data back was just to plot all of these reads in the genome of Salmonella, and then we can look at the insertions present in the input and compare that to the output. And we wanted to ask if we got selection for known virulence genes, like you would expect in any kind of TN-seq experiment. And so what I'm showing you here is a 27 kilobase snapshot of the genome, in which we have the genes for Salmonella pathogenicity island 2. So each of these little pink bars here represents different genes in this pathogenicity island. And just as a reminder, this SPY2 is induced by FOPQ upon entry into macrophages and is required for intracellular survival. And so to interpret the data here, so on the top in the green, we have the wild type input population. So each one of these peaks represents reads that are matching to individual transposon insertions throughout this region of the genome in our input pool. And then we have our 3 output groups, 1, 2, and 3. And so you can see that most of these peaks that are present in the input pool disappear in our output pool. And that tells us that those mutants did not survive our infection conditions, which is what we would expect in this case, since we know that these genes are important for survival for systemic infection. And what's interesting is actually, if you go in and look at these peaks that are still present within this region, so these 2 genes are actually secreted effector proteins that get secreted via the type 3 secretion system into the macrophage. And while they've been shown to have a role in virulence, some groups have shown that they're not absolutely required. So it's not surprising that we still might identify insertions in those genes in our output population. So next we needed to move on to the actual data analysis, because as I mentioned, the key part of this experiment is identifying genes with different phenotypes in a wild type background compared to a SOD-C background. And so the way we did our data analysis was we collaborated with the HPC Bio group for help with the initial statistical analysis of our data. And after that point, we ended up with a list of about 600 genes that had a phenotype of interest, meaning a potential genetic interaction with SOD-C1. And to narrow that list down further, what we did was we used IGV, which is the program I showed you the data in on the last slide. And Jim and I plotted all these insertions in the genome, and then he started at one end, I started at the other, and we marched towards the middle. And for each gene, we looked at the location, the fold change, the significance, and then also layered on our understanding of the biology. So a gene worked as part of a functional complex or as part of an operon. We looked to see that if genes in that complex or operon had similar phenotypes, which gave us more confidence that that might be a real genetic interaction. And we ranked these genes on a scale of 1 to 3, with 1 being our top priority for follow-up. And at that point, we were left with a list of about 95 genes ranked with 1 that were our top priority for follow-up. And so here's the genetic interaction breakdown of that data. 29 of those genes had putative synthetic interactions. So one example of that is YGBD, which had no phenotype in a wild type background and was significantly attenuated in a SOD-C mutant background. 64 of those had putative epistatic interactions. An example is YIDA, which was significantly attenuated in a wild type background, but had no phenotype in the SOD-C background. And then finally, 2 of those had a suppressor phenotype. I didn't introduce the suppressor phenotype earlier in my talk, but the way this can be explained is that insertions in this gene had no phenotype in a wild type background and actually had significantly increased virulence relative to the rest of the population in the SOD-C background. So suppressor suggesting that this is suppressing the phenotype of a SOD-C1 defect. The next step in this project was then to verify these genetic interactions. And the way we wanted to do that was to use our standard 1-to-1 competition assay. Unfortunately, with the way these mutants were generated, it was randomly in pools. And so we had to go back and recreate the individual mutants in each of these genes. And then we did that using the lambda-red system, and then moved those mutations into a wild type and SOD background and tested them in our competition assay against the parrot strain. And I'd like to acknowledge an undergrad, Andy Wu, who worked with me on this part of the project. He joined the lab right when I was starting this and got the fun job of generating all these mutants using lambda-red. Became very efficient and was really helpful for working on this part of my project. So far I can report that we've actually confirmed 3 novel genetic interactions with SOD1. Those are shown in the table here. So first we have YTFL. YTFL in competition against wild type is about 7-fold attenuated, whereas it has no phenotype in a SOD1 background competed against SOD. This is an epistatic interaction. HCP and YLIG both had synthetic phenotypes, meaning that they had no significant phenotype in a wild type background, but were significantly attenuated in a SOD1 mutant background competed against SOD1. And so in the interest of time, for the purpose of today's talk, I'm just going to focus on YTFL. So I think that's our most interesting result thus far and has a lot of exciting data to move forward with in the lab. So on the top here, I'm showing you the genetic locus of YTFL. So it sits nearby some other YTF genes, including YTFJ, YTFK, and is downstream of MSRA, methionine sulfoxide reductase. And so the question I'm sure everybody wants to know is, what is the function of YTFL? And as you might have guessed by its name of a Y gene, we don't know what the function of YTFL is, unfortunately. It's never the easy way, of course, but based on predictions, we believe YTFL is a hypothetical intermembrane protein with a cytosolic C terminal. So the simple interpretation, again, of an epistatic interaction is that YTFL is a target of superoxide, and SOD1 functions to defend YTFL's unknown function by protecting and dismutating superoxide. One question we wanted to ask when we initially got this data back was, is this phenotype specific to an in vivo infection, which suggests it's related to virulence, or is this just a general growth defect that we might be able to repeat in the lab? And so if we look at the same competition assays, except this time instead of using mice, we just grow these competitions overnight in lab media, we see there's no significant phenotype suggesting that this is specific to our in vivo infection model. Now, I got this data about the time that I had to transition back into my medical school curriculum, and I did some preliminary experiments trying to understand if other genes nearby YTFL also had genetic interactions. I looked at YTFK, YTFJ, and MSRA, and didn't identify anything. So I'm not showing that specific data today. But while I was back doing my medical school curriculum, some other members in the lab were working on function and understanding of YTFL. I'd like to share some of that data with you today because I think it makes this story more interesting and more exciting about why YTFL is a really interesting discovery from this team's experiment. And so here, I'd like to talk a little bit about regulation. And so we know that Kaka, a postdoc in the lab, has been studying the regulation of YTFL, and she's shown that YTFL is regulated by FOP both in vitro and in vivo. So YTFL is induced almost 20-fold in vivo by FOP. And I've mentioned before, but FOP is part of the two-component system important for induction of virulence and systemic survival. And it's interesting that FOP also is a regulator of SOD1. And through additional work, she showed that this regulation actually occurs at the MSRA promoter and is via read-through into YTFL. And other work in the lab, Yumi, a really talented postdoc, has found that YTFL is actually sensitive to polyamines. This is interesting. Polyamines have been studied from both a host and pathogen side. It's been shown to be involved in regulation of gene expression in hosts related to nitric oxide, but also in regulating the types and the differentiation of immune cells, which can affect virulence. And from a pathogen side, in Salmonella, polyamines have actually been reported to be involved in regulation of SPY1 and SPY2, and been shown to be required for intracellular survival. So I think it'll be really interesting to follow up more on this data. And then the last bullet point here, Ruhala, another graduate student in the lab, has been studying this potential interactions of YTFL with YTFM and YTFN. And he's looking at this in light of YTFM and N might function like a BAM complex, and has some genetic interaction data with that. So I think that'll be interesting to really follow up on as well. Now, I would be remiss if I didn't mention that this experiment didn't come without its challenges. While we all like to think that science is going to work perfectly on the first try every time, we definitely ran into some unexpected bumps in the road in this data, and it was an inherently challenging experiment both in size and complexity. And the issue with moving forward with publishing the screen as is that our confirmation rate has been much lower than we expected. And so I showed you the 3 genetic interactions that we've discovered so far, but for every one of those, there's several other genes that we've tested to try to confirm and don't have the predicted phenotype. And we think that's because there's, for an unknown reason, a high amount of background noise in our data. And as Jim mentioned in his introduction, Yumi, a postdoc in the lab, has been working with Dr. Jensen to reanalyze this data. And so working with Dr. Jensen, Yumi has created a new method to prepare these sequencing libraries. She redid the sequencing a few weeks ago, and the initial results from this are really promising. There seems to be a lot less background noise than in our initial analysis. And so I'm looking forward to when we can get back into the lab and actually start to confirm some of this data and hopefully have a cleaner analysis this time around. So stay tuned for those results. Now I'd like to spend a few minutes talking about the RNA-seq experiment that we use to complement our differential T-and-seq experiment. And so our hypothesis for the RNA-seq experiment was that by comparing the transcriptional response of wild type and a SOD1 mutant during macrophage infection, when the bacteria are responding to the oxidative burst, we might be able to identify a transcriptional signal that gives us insight into the nature of damage caused by superoxide. And this experiment is something I've done in collaboration with Katka. She was the one that did this initial infection and RNA isolation. And the way this was set up was we took 3 independent cultures of wild type and of a SOD1, SOD2 mutant, initiated tissue culture macrophage infections. And then after 2 hours, which is the time point based on the literature and some preliminary experiments, we thought the bacteria might be responding to the oxidative burst. Katka isolated RNA from those, and then we submitted that for sequencing. And to analyze our sequencing data, we used Rockhopper, which is a freely available bacterial RNA-seq analysis platform online that was developed by the Vanderpool lab as part of a collaboration they had worked on. And our results from that was we saw 110 genes that had at least 2-fold or greater differential expression during in vivo infection. And so 16 of those had relatively increased expression in SOD1 compared to wild type, but the vast majority, 94 of them, had decreased induction in a SOD1 background. And what was interesting about this and really caught our attention that was unexpected was there was a lot of virulence genes that were present in this list of 94 genes. And here I'm showing you a select number of those genes that had decreased induction in SOD1 compared to wild type. So in each of the boxes, I've listed the genes from our data, and then in parentheses, this number is the log 2-fold change. And what we jumped out at us was that a lot of these genes fell into regulons of 3 known virulence regulators. So FOP, I've mentioned several times, SSRB is encoded with SPY2, and it's important for activating the type 3 secretion system apparatus and effector proteins. And then SLY-A has a lot of overlap in expression with FOP and has been shown to also be important for systemic infection. And so our take-home point from this data was that, which was really unexpected, was that for some unknown reason, which we're still trying to figure out, it seems that in a SOD1 mutant, there's decreased induction of genes that are important for survival systemically. And so this really resulted in almost a paradigm shift in how we think about and interpret our SOD1 virulence phenotype. And so based on this data, we think that there's altered gene expression on a more global level, resulting in induced expression of virulence genes in SOD1 relative to wild type. Whether there's also an alteration in the host response to Salmonella infection is not specifically clear, but is something we're going to follow up on. And so how do we combine these 2 results from the differential T-and-seq experiment and the RNA-seq experiment? So in the differential T-and-seq experiment, I told you that we identified 3 novel genetic interactions and focused on discussing about YTFL, which in a simple interpretation can be thought of as the target of superoxide. Then in the RNA-seq experiment, now I'm saying that it seems like there's global alteration in gene expression. So these at face value seem to be 2 results that don't pair well together. But actually, I think it's a really exciting result and that goes well together and will be interesting to follow up on. And the way that I explain that is if you think about the epistatic interaction with YTFL, the two ways to interpret the data are that, as I mentioned, YTFL is directly inhibited by superoxide, or the alternative hypothesis is that in this in the altered gene expression in a SOD1 mutant background, the function of YTFL is no longer required. And so both of these interpretations would result in an epistatic interaction of our TN-seq data. And so we'll have to do continued work in the lab to really try to differentiate between these and try to figure out what's going on with YTFL and this potential altered environment. So for future directions from my work, I've mentioned a lot of this already throughout my talk, but would just like to highlight some of these things that are going to continue to move forward in the lab after I've graduated. And for YTFL specifically, Kaka continues to work on various aspects of the regulation. Ruhala's project looking at YTFM and YTFN, he's shown that YTFM is actually also epistatic with SOD1 and epistatic to YTFL. So it'll be interesting to follow where that data goes. And then also the in vitro phenotype that Yumi has been working on with the polyamines is a really exciting story that I think you'll hear more about relatively soon, hopefully. And then in regards to the differential T-and-seq, we're at the point where we are doing a statistical analysis and comparing these new lists from this new library preparation, thanks to a big effort by Yumi and lots of helpful input from Dr. Jensen. And so I'm excited that we'll be able to return to the labs in the near future and start following up on some of the confirmation of this new gene list, and hopefully have a little bit cleaner analysis this time around. And then for our RNA-seq data, again, Kaka is going to work on looking at the transcriptional response from a host perspective in response to SOD1 mutant or wild type. And then also she's working on confirming these virulence genes and to see if they actually have decreased induction in a SOD1 background, and has some interesting preliminary competition data for that. And so thank you all for tuning in to hear some of my work today. This definitely was not a solo effort, but the result of hard work from a lot of people. And there's a lot of people that have helped me along the way that I'd like to acknowledge at this point. Before I do that, for those of you watching on the live stream, if you have any questions, if you put those in the comments right now, I'll be able to check that when I'm done. With the acknowledgements, there's a little bit of a delay. So if you have any questions, please put those in now. Otherwise, thanks for tuning in. And first, big thanks to Jim. I couldn't have imagined a better thesis advisor and mentor. It's really been a great experience in the lab. It was challenging at times, but I knew I could always come into your office and we would get things going on the right track. And it's been really rewarding experience for me. I've learned so many lessons that I'll continue to use in my career going forward. And I really appreciate all the opportunities that you've given me over the years. To all the members of the SLAO Lab that I've worked with over the years, many of them, I mentioned their specific contributions today. Others that I didn't mention have helped in other projects, either directly or indirectly, but just as importantly, it's been really fun to show up to the lab every day and work with all of you. So thanks for making that a really fun and great time. I also like to thank my thesis committee for all of their helpful advice over the years and input on my projects. And an additional thanks to Dr. Imlay. He's the one that personally taught me how to do the SOD assays that I showed you earlier on in my talk, and also to his lab for letting me invade some of their lab space to do those assays. Dr. Alvaro Hernandez and the HPC Bio Group were instrumental in getting our initial sequencing and analysis up and running. And then Dr. Jensen has put in a lot of work to help us try to understand our data better and really help guide us in the right direction for this T-and-seq reanalysis. And again, Yumi has been leading the charge for that in the lab and been doing a really terrific job. I'd also like to thank some people from the micro office and MCB office. So Diane, Deb, and Shauna, we all know the ones that really keep everything, the wheels spinning in the department. And they've been really great to work with over the years. Also, everyone involved with the Medical Scholars Program. It's been a long journey, but it's really been a pleasure to be a part of. And I've really enjoyed it. And then also to my various funding sources I've had over the years. And since this is my thesis, I'd also like to take a minute or two to thank some people that have helped me, not just inside the lab, but also outside the lab. So I've made a lot of really great friends during my time in Champaign-Urbana and created a lot of really great memories that I'm going to look back fondly at for many years to come. So thanks to all of you that have played any part in some of that. And I'd specifically like to say thanks to my family. So my parents who are tuning in today from Wisconsin to watch this virtually. I wouldn't be where I am today without all of the help and support they provided for me over the years. Although it seemed like I may never graduate, I knew I always had their support, and it was really helpful to keep me going. And also my sister, her husband Kevin, and my awesome niece Zella. I can't wait to see you guys sometime soon and celebrate, hopefully in person eventually. And then most of you all already know my wife, Courtney. She doesn't really need an introduction to this audience. She was also a PhD graduate from this program and will always be the original Dr. Fenlon in our family. And her, and this is our dog Tully, have just been instrumental in keeping me going every day. They always put a smile on my face. And their support has been crucial to getting me through both medical and graduate school. So I'm excited about all our adventures to come, which start with moving to Utah for residency tomorrow. And then also when I married Courtney, I acquired some new family, and I've had some great adventures with them already, and I'm excited to be able to see all of them and celebrate as well. And you can see on the bottom here, this is our dog Tully, and he's done a really good job of adjusting to the isolate at home. So with that, I'd be happy to take any questions that anybody might have. All right.